In this video lecture, I'm going to talk about celiac disease and diabetes mellitus type 1 association. And as you know that celiac disease is an autoimmune disease caused by ingestion of gluten in genetically susceptible individual and characterized by auto antibodies. However, also besides the antibodies, it is also T cell disease. So celiac disease is a common cause of chronic malabsorption in children and is characterized by mucosal damage, as can be seen here. The villi in gluten sensitive enteropathy or celiac disease get atrophied as compared to normal villi. It is actually the gluten is present in wheat, and this association between celiac disease and diabetes mellitus type 1 in children has been reported in many studies, and it is around 1.3% to 12% of children with diabetes mellitus may suffer from celiac disease. So up to 12% of children with diabetes mellitus type 1 may suffer from celiac disease. And this association has been supported by the reduced prevalence of type 1 diabetes mellitus after gluten deprivation in patients with celiac disease. So basically, genetic factors play a major role in this association between celiac disease and diabetes mellitus type 1. So both celiac disease and diabetes mellitus type 1 are associated with HLA, DQ2, and HLA DQ8. So these are present in the majority of patients with celiac disease and in patients with diabetes mellitus type 1. So since 2 to 3% of celiac patients have selective deficiency of IgA, however, recent new tests have been introduced, and these tests detect actually IgG antibodies, and one of these tests is anti-tissue transglutaminase antibodies or anti-TTG. So we know that celiac disease patients, they have selective deficiency. Not all of them actually, 2 to 3% of them, they have selective deficiency of IgA. So the classical test, for example, anti-gliadin or anti-endometrial antibodies, this depends on detection of IgA may become negative, while the patient is truly a case of celiac disease. So, recent investigations or test by detection of anti-tissue transglutaminase antibodies as antibodies of anti-tissue transglutaminase antibodies. They are of IgG. So still you can diagnose celiac disease in IgA deficient patients. So actually study by Bolad and others reveals that the association between celiac disease and diabetes mellitus type 1, it is around 10% among Sudanese children, and actually this finding by Bolad and others among Sudanese children with type 1 diabetes mellitus are attention grabbing, because celiac disease is expected to reduce weight and consequently body mass index. So we advise that all children with diabetes mellitus type 1 should be investigated for celiac disease, as the association is a bit high. But however, despite this association, the role of dietary gluten development in development of type 1 diabetes mellitus, and the potential effect of removing gluten from the diet of patients with type 1 diabetes mellitus are still debatable. So this association, it has been shown in several studies, but however, the removal of gluten from food of patients with diabetes mellitus type 1, and just to avoid this association, as I mentioned, they are still debatable. So, as can be seen here in this drawing, that this is the gluten. It is formed of these red circles. This is gliadin, and these reds are glutenin. So, however, gliadin can cross the mucosal lining of the intestine to lamina propria, and as can be seen here, within the epithelial cells, there are some these epithelial cells from patients with celiac disease. So one can see here CD8 or T cytotoxic, and actually these play a role in immunopathogenesis of celiac disease and destruction of the mucosa, as they produce granzymes, perforin and interferon gamma, and this will lead to tissue damage and inflammation, and it will lead also to recruitment of this interferon gamma, can recruit macrophages to the area. Actually, these gliadin peptides, when they translocated to the lamina propria, and when they crossed these epithelial cells, they will be modified by tissue transglutaminase 2. This is an enzyme, and this modified structure will be engulfed or phagocytosed by antigen presenting cell, maybe macrophages. So this antigen presenting cell, they present peptides from these both gliadin and tissue transglutaminase to specific T cells that produce these antigens, and specific T helper cells get activated, and they produce interleukin number 2 and interferon gamma, most probably this T helper one. And as you know that interferon gamma recruit more macrophages, and interleukin number 2 activates B cell. So now B cells enter the game and get activated by this, and also it is an antigen presenting cell, so they can take these molecules, gliadin together with transglutaminase, process them and present them to T helper that most probably here it is T helper number 2, and it helps T helper produce interleukin number 4, which is a growth factor for B cell. So B cells differentiate into memory B cell and plasma cells, and plasma cells will produce anti-gliadin antibodies and anti-tissue transglutaminase antibodies, or anti-TTG. So these antibodies, when they bind to specific antigens, whether it is gliadin or transglutaminase, this will also further deteriorate the condition by activating an inflammatory cascade by the complement system, and this will lead to more destruction of the epithelium or the mucosa, and as can be seen here, this atrophy, this epithelium and the mucosa get atrophied, and the villi here actually get atrophied as compared to villi in the normal mucosa. Thank you very much, Professor Ahmed Boulad.